Hi, so we're on our third unit, biology. See, this is the first time I've had no one show up to the lessons, and it's not even funny, but whatever. All right. So honestly, bio is like my worst science. Maybe chem is, I don't know, one of the two. Since bio is mostly memorization, it doesn't require application with like physics where you had formulas, and then chemistry is like, I don't know, chemistry is chemistry. That doesn't matter. Anyway, for the rest of pilot prep science 10 lessons, you, or at least for the ones I'm teaching, you don't need to bother to take notes anymore. All right, if you need something, just go back to the slides and then you can find what you're looking for, which you should have access to in the Google Drive. Bio tips. So the key to acing bio, even though I'm not that good at it, is memorization. You can do this through active recall, which you may have heard of. It's a pretty well-known study strategy that you should use for honestly all your classes. It's like, think of it as flashcards. I can't be bothered to just explain it, but just like Google it. And then the next tip is use methods to remember the stuff you learn in bio, like acronyms or songs, etc. You know, yeah. Okay, so our first topic we're going to address is microscopes. So a term we need to know for the microscopes topic is resolution, which is the ability to distinguish between two structures that are very close together. It can be increased with contrast and staining. So for example, if you look down here, we're looking at the same image, but think of it as this one's under low resolution and under high resolution. Going back to my definition, the ability to distinguish between two structures. Here you can see, even though both images are showing two separate structures, this low resolution doesn't allow you to distinguish that they're two separate structures. But the higher resolution is like, oh, I can see that, they're two white dots. And then going to contrast and staining, how do I explain that? I think I explained it farther on the next slide, on later slides. All right, contrast and staining, there we go. You can increase contrast by manipulating light, which is the diaphragm of the microscope right here. If you've ever used a microscope in school, you can like kind of scroll this thing, move this thing to adjust the amount of light going in. Staining is used to increase the contrast, to help increase contrast, and therefore it increases resolution. Staining is just like adding color to cells. It's like, think of it as adding food dye to cells, but don't actually add food dye. Because the reason staining works is because unstained cells, they're generally colorless, so they usually make black and white images, which makes parts harder to distinguish, as in like different parts of the cell. Moving on, the electron microscope. The electron microscope was invented by James Hiller and Albert Prebys of University of Toronto. I know, right? Canadians in science history. That's actually amazing. The electron microscope uses beams of electrons instead of light waves, like the light microscope, which is this thing. And the first electron microscope made was the transmission electron microscope, or I write TEM for short. It uses beams of electrons again, and then passes. Wait, let me reread this. Beam of electron passes through thin, stained, fixed tissue. There we go. Exactly what it sounds like. It creates black and white images showing internal composition. So here on the right, we have an electron microscope, and then this is the image that you would get with a transmission electron microscope. So again, the electron beam passes through the object you're looking at. This is important to know. Next, we have the scanning electron microscope, or SEM for short. When you look at specimens under the SEM, they have to be fixed and covered with dense materials that reflects electrons, so such as gold. And the scanning electron microscope shows the surface composition of the object you're looking at. So here on the left. As you can see, these are the same things, but this is under the transmission electron microscope where you can see the inside composition. And then this is under the scanning electron microscope, where you see the outside structure. And again, electron microscopes are like pricey. So your high schools won't have this thing, but universities, if you enter biology or some science major, you will have access to an electron microscope. Also, you need to know the difference between like a transmission electron microscope and a scanning electron microscope. Moving on. Cell size. This is the formula for cell size. Cell size equals FOV over fit number. FOV stands for field of view, which is how much you see when you're looking through the microscope. It's measured by the width or diameter of the area you see. Fit number is the number of times the cell fits across the field of view. It's usually rounded to a whole number. And this is a magnification formula where total magnification, so the magnification you're using is equal to the ocular lens magnification, which is this, the magnification of this tube thing, multiplied by the objective lens magnification, which is the stuff down here on the light microscope. Ocular lens are almost always have a magnification of 10 times. And then the objective lens, it can be 4 times, 10 times, or 40 times. So if I were to give an example of total magnification, let's say I'm looking through my ocular lens while using an objective lens of 4 times power. Using this formula, ocular lens magnification times objective lens magnification, it'd be 10 times 4, which is 40. So my total magnification would be 40 times. An example of a cell size calculation, let's see. Let's say I have a field of view of why can't I write? Please. Oh, there we go. 4 nanometers, which is like tiny, I think. And then the cell, let's see, here's my FOV. This is what I see when I look through the microscope. And this is my cell. To find the fit number, this cell, like kind of, let's see, how many of that cell would fit across the screen? This is so not to scale. 1, 2, 3, 4, 1, 2, 3, 4, 5. Let's say 5, okay? Let's say 5. So the fit number is 5. The number of times the cell I'm looking at fits across the screen. So FOV 4 nanometers divided by the fit number, which is 5, equals 4/5 of a nanometer. And that's the cell size. That's the size of the cell you're looking at. Here's my eraser. Let's see. Let's get rid of this stuff. Next. So something you need to do maybe is, this might be tested on. Like in school, they might ask you to know the difference between the microscopes. So a light microscope, again, this thing, also referred to as a compound light microscope. They are cheaper, so therefore affordable by high schools and junior highs under the CBE funding system. They are portable since they're pretty small and they're light. You can look at live specimens under the light microscope. So you can, put like living bacteria under the microscope and look at it, how it moves and such. You can see the natural colors of a specimen under the light microscope. So if you put in a blue stain under the microscope and you look through it, you'll see blue. It is optional to stain things under the light microscope. So adding, again, my metaphor was food dye. Electron microscope, they have a much greater magnification, so you can see things like many times bigger with a greater resolution, much greater resolution actually. But you can only look at dead specimens, and electron microscopes only produce black and white images. Example 1. You have a cell 5. This is the unit for micrometers. You're going to see this unit a lot in grade 10 science and up. So a cell that's 5 micrometers big and it fits across the screen like 20 times. What is the FOV? I'll give you guys like 20 seconds for those of you watching from behind the screen to complete that, and then I'll do the question. Oh wait, I should probably show you guys the formula. I'm actually dumb. Here's the formula. Since I told you guys to not take notes, you probably don't remember the formula. Cell size equals FOV over fit number. I should write that. FOV over fit number. And I will now solve the question. So we're looking for the FOV, and the equation itself is like size equals FOV over fit number. And we are looking for FOV. So let's rearrange this equation so we can find FOV. To do that, we'll multiply both sides by fit number. This is taking a while. Fit number. Let's write number. Can't be bothered to write fit number. And then that makes, then this cancels out. These two cancel out, resulting in a formula like this, where size multiplied by fit number equals the FOV. So let's see. The size is 5 micrometers. Micrometer symbol is kind of, that's bad. That's such a horribly drawn symbol. And the fit number is 20. So 5 times 20 is like 100, I think. 100. And then the unit is micrometers. So it's 100 micrometers. That is your field of view. This writing is hardly legible. Anyway, what does FOV stand for? What is FOV? Well, field of view, the width or the diameter of what you see when you look through a microscope. So you shift your objective lens to high power on your microscope. You see that the high power lens has a magnification of 40 times. What is the total magnification you'll see when looking through the lens? As a reminder, ocular lens has a magnification of 10 times, and the formula for total magnification is magnification of ocular lens multiplied by the objective lens. Go solve. I'll now show you how to do it. Okay. So total magnification is the ocular lens magnification, which in this case is, or it usually is 10 times. 10 multiplied by the objective lens magnification, which is here, 40 times. So the magnification you'll see when you look through the microscope will be 400 times. Beautiful. Moving on. Cell theory. Let's erase this. So, cell theory. Yeah, it is what it is. Cell theory. So there are 3 parts to cell theory, which is number 1, all living things are made of cells. 2, cells are the basic unit of structure and function for all organisms, which another way to put it is just cells are the smallest unit of life. And then number 3, all cells come from pre-existing cells. This you might be tested on in school, so I suggest memorizing cell theory when you're in Science 10. All right, cell membrane. So the cell membrane is a part of the cell. See if I have an example of a cell image. Here's my cell image examples. There we go. Cell membrane is just like, here, you know what? The skin of a cell, there. That's a better metaphor. Skin of a cell. Oh, I wrote that there. It's also known as a plasma membrane, and although it's the skin of an animal cell, for the plant cell, it's actually the inner skin, because the plant cell kind of has two skins. The cell membrane also controls what gets in and out of the cell. It is also semi-permeable, which just means some things can get in and out, some things can't. As in the cell membrane, let's see. It's like, how do I explain that? I hope you get what I mean. Semi-permeable. Some things can go into your cell. The cell membrane blocks other things like bacteria from getting into your cell, or certain viruses sometimes. I know. Semi-permeable. Cell membrane only lets in the things it wants in the cell. The things it doesn't want, it blocks out, usually. So the cell is an open system since it lets things come in and out of the cell. The cell membrane is made of a phospholipid bilayer, which if you look at here, let's make this big. Phospholipid bilayer. See these dots? See these dot things? Those are phosphates. And see these string-like things? Those are lipids or lipid tails. And then these things combine together, one phosphate with two lipid tails creates a phospholipid. And then there's one layer of phospholipid, and that's another layer of phospholipid. So we call it a phospholipid bilayer. Okay, the model we most commonly use or most commonly accepted for the cell membrane is a fluid mosaic model, which is this model. There are other models, older models of people trying to guess what the cell membrane looks like. You can search them up if you want. I don't know. Next, cell wall. It is a rigid frame on a plant cell which provides strength and support. Yeah, it contains cellulose, which is just a fiber, and it only exists in plants, so you will not find a cell wall in animal cells, and this is important to know. Cell walls, they exist for added structure or stability in plants since plants don't have bones. So think of it this way. If you step on a human, they'll die. Like if you had a giant foot and you crush that human, they'll probably die. But if you had a foot and then you step on like grass or plants, they're completely fine. They'll come back up. They'll be crushed, but they'll come back up. They'll be fine. This is what cell wall does. It just gives them structure that makes them more resistant to being stepped on than animals. So the nucleus, it is the brain of the cell. It like controls what the cell does. It contains the genetic material of the cell, which is the DNA or deoxyribonucleic acid for those of you interested. This is an image of a nucleus, like this big circle thing. This is just stuff inside the nucleus. The nucleus is, I think, the biggest organelle in the cell. So when you're looking under a microscope, you'll usually able to see a compound light microscope. You'll usually be able to see the nucleus. As for other organelles, when you're looking under compound light microscopes, they're very hard to see. So yeah, I'll explain more on that later. Nucleoplasm, it is the, you'll learn what a cytoplasm is later. I don't know why I used the word here before I defined it for you guys, but cytoplasm is just like the juice of the liquid inside the nucleus. So this light purple stuff, just the watery stuff inside the nucleus. Nucleolus, it's like a mini nucleus inside the nucleus. It's a knot of chromatin, which is just a type of DNA. And in the nucleolus, ribosomes are produced. And ribosomes make proteins. So this tiny circle inside this nucleus is a nucleolus. And as you can see, all this stringy stuff, that's chromatin. So it's just a knot of chromatin. Cytoplasm. It holds cellular organelles in place and contains cell nutrients. It's mostly made of water and it allows the movement of like stuff within your cell. Yeah. Mitochondria. Okay, I don't know if you guys have heard of this, but there's a famous quote called the why am I scrolled so far in? What is happening? Okay, whatever. The mitochondria is the powerhouse of the cell. You guys might have heard of this before. It's a pretty famous quote that almost every school teaches in Science 10. You'll find, eventually at some point you'll start seeing them on memes online, not gonna lie. So the mitochondria, it produces energy by converting chemical energy into fuel, which is the fuel in our body is ATP, or adenosine triphosphate. This is important, ATP. The folds in the mitochondria, so these like wrinkly things in the mitochondria, as you can see, there's like stuff sticking out here. In the mitochondria, this is so there is more surface area. And biology, things in bio love surface area. Tony was supposed to back me up on this, but he's not here. Anyway, a lot of structures in biology, they will go out of their way to maximize surface area. This allows for more cellular or more efficient cellular respiration to happen, which is the process of energy conversion from chemical to fuel in the body. Let's see. Yeah. Chloroplasts. These are only in plant cells, and they are what gives the plant the green, why plants are green, typically. They produce energy, but a different one from the mitochondria. So in the mitochondria, mitochondria uses chemical energy to produce ATP, but chloroplasts, they change sunlight into food through, take a guess at the process. You guys have all learned this since like what, grade 2? Photosynthesis. And here is an image of a chloroplast. It is green and looks like a bean. Yeah. Whoa. Whoa. What is happening? Ribosomes. They are. What is happening? Very small. I mentioned it before, they make proteins. And if you remember, they are made, or synthesized is a fancy word for made, synthesized in the nucleus. And ribosomes synthesize proteins. There, you can now impress your bio teacher. Next, the endoplasmic reticulum. I don't know why my screen is like this. What did I do? What did I do? I'll figure that out during the break. Endoplasmic reticulum. There are two kinds of endoplasmic reticulum. There's the RER, which is the rough endoplasmic reticulum, and it is a pathway to transport proteins, aka ribosomes. So after your ribosomes are made in your nucleolus, they're transported. This is the rough endoplasmic reticulum in the bottom right corner. After they're made in the nucleus, they're sent to your rough endoplasmic reticulum, and then your ribosomes just travel around your RER to get to where they need to be. My teacher remembers them by calling them a highway for proteins. Next is second type, SER, or the smooth endoplasmic reticulum. They are associated with fat and oil production. Now, I should probably explain why they're called rough ER and smooth ER. Since the rough ER, you see here in the image, has ribosomes on it, which are the red dots, it has a rough surface. So the smooth ER, it doesn't have ribosomes on it, so it has a smooth surface. Now, the RER is typically located right by the nucleus of the cell, and then SER is like located just outside of the RER. I break 10-minute break, but there's no point in making a break because there's no one here. I'm gonna use this break to fix my screen. Bye-bye. See you in 10 minutes for me. Hello there. So unfortunately for me, my break is over. Oh yeah, I wanted to talk about Halloween. Happy Halloween, guys. Moving on. Golgi apparatus. So think of it as a post office. And now going back to like the second slide when I talked about using metaphors or stuff to help you recall stuff. This is an example of a way to recall something. Now, it is important to remember like the functions of the organelles I am teaching. Since you will be in school test, you will be tested on this. Anyway, the Golgi apparatus is usually located close to the endoplasmic reticulum. So if you look here, right beside the RER and SER, it's the Golgi apparatus. Yeah. The role of the Golgi apparatus is to package and ship out proteins. So again, think of it as a post office. And we see here, this is the Golgi apparatus, and this is it packaging and shipping out proteins. The stuff is coming off of the Golgi apparatus into balls, and it floats away around the cell. All like random sac balls, oh my god, all random sacs in the cell, these are all called vesicles, these little circle things. Lysosome. So think of it as like Lysol, since it digests wastes within the cell with digestive enzymes. So here we have the cell, and this thing, it's a lysosome. It cleans up the cell by digesting waste, and it can destroy invading viruses by making the cell self-destruct, also known as cell suicide. But usually the role of lysosome is just to digest waste. Moving on. Vacuoles. They store food and water. In plant cells, vacuoles are huge. Usually vacuoles in plant cells, they only have one. But then in animals, we use it, or like humans, the vacuoles in human cells, for example, they're much smaller, and we would have many vacuoles in our cells compared to plants which just have one big one, also known as a large central vacuole. Next up, centrioles. These are only in animal cells. They take care of mitosis, which is cell division, where one cell becomes two cells, which is cell reproduction. I think they look like pasta. I just wanted to put that out there. And centrioles are also usually found close to the nucleus. Okay. A really common test question you're going to have is to compare and contrast plant and animal cells. So here's the answer on the next slide. You can, if you memorize this and then you get that test question about comparing and contrasting, you can like copy and paste this, and then you probably get almost full marks, close to full marks, maybe full marks. So firstly, the difference, this is just the difference, contrast of the two cells, where in animal cells, they have, again, multiple small vacuoles. They contain centrioles, and they are rounder in shape, or just not square, not boxed in shape. So down here we have an amoeba proteus. As you can see, it is oddly shaped. And I can't find the nucleus on here. But plant cells, they have large central vacuoles. Oh, they're usually green. So yeah, you guys know that plants are usually green because, again, of the chloroplasts. By the way, chloroplasts have like a green pigment in them, and that's why the whole plant's usually green. Plants have a cell wall, which give it its boxy shape. So if you look here, which is the onion skin cell, some of you might have actually already looked at this stuff in like junior high science. You can see that its shape is like rectangular or boxy compared to this amoeba. And again, it contains chloroplasts. So if we look at these diagrams, again, animal cells is rounder, and then the plant cell because of cell wall giving it more rigid structure, it makes the cell more boxy. So I said before that the cell membrane is like the skin of the cell. So here we have, this is the cell membrane of the animal cell, just the skin. And then for the plant cells, you see this thick stuff, that is the cell wall, but this orange-ish, brownish lining right inside of the cell wall is the cell membrane. Also, I mentioned before that the nucleus. Yes, the nucleus, the organelle within a cell, is the one you'll see when you look through compound light microscopes, and the biggest organelle, aside the cell membrane / cell wall. This purple dot on each of these cells, that is the nucleus. All the other organelles, like chloroplasts, they're too tiny to be seen through this microscope. And then looking back at the vacuoles, you can see here, this is a plant vacuole. It's huge, and there's only one. Whereas in animal cells, the vacuole looks like this is a vacuole. They are small, and we have more than one. There should be quite a few in here. Okay, moving on. The chemical composition of cell structures. So cells, structures that contain lipids, which is fats and oils. These are terms, by the way, that you will need to know. Lipids, fats, and oils, where oil is just plant fat. Carbs are sugars, starches, and cellulose, where if you decide to keep going with bio and take biotin, you'll learn that sugars, starches, and cellulose are like the same thing, but not. Starches are essentially a bunch of sugars, but combined, and the cellulose is a bunch of sugars / starches combined. Protein. Examples of protein are muscle fibers and enzymes. And then we have nucleic acids, which is DNA and RNA. Deoxyribonucleic acid and something nucleic acid. The last thing is trace elements, also known as micronutrients. Some teachers call it differently. These are just nutrients your body only needs in small amounts, like magnesium. So when you're eating, you don't need to eat a lot of magnesium. Unlike calcium, your body needs a lot of calcium. Drink milk, guys. History. So firstly, one belief within history about life and existence is known as spontaneous generation, also known as abiogenesis, which is just the belief that life appeared spontaneously or out of nowhere. It was believed until around 1859. And the reason people believed this was because if you left like food out for a few days and then came back, mold or maggots, worms or stuff, all right, they'd start growing on that piece of food you've left out for days. And it just seemed like this life came out of nowhere. It spontaneously generated. But then scientist Reddy proved it wrong by where he left meat out for a few days, but he covered it. So let's say he wrapped it in plastic or something, which probably did not exist at the time, but you get the idea. He covered it up, he left it for a few days, he came back and he's like, no maggots grew on this piece of meat. Therefore, spontaneous generation is fake. Now, if my titling here is not right, so where does spontaneous generation come from? It was believed that spontaneous generation came from life force. This was influenced by the fact that most people all the time were all like Catholic. So basically, scientist Needham suggested there was a life force that caused spontaneous generation, and he believed sterilization took out this life force. So if you wiped something down with alcohol, if you wiped something down with alcohol, it was cleaned. And there would be no life force left in it. But then Pasteur proved it wrong through pasteurization. And that is where the name comes from. Pasteurizing, also known as boiling stuff, when you pasteurize something, it didn't stop things from growing in there. So if you boiled milk and then left it out for a few days, mold. Does mold grow on milk? I don't know. You get my idea. If, let's say, mold grows on milk, after you boiled stuff and left it out, mold will still start growing on the milk. So boiling stuff, or sterilization, didn't stop things from growing, so there is no such thing as a life force. Now, some influential people of history in biology. Aristotle. You've all heard of him, this Greek philosopher man. He was the first to come up with the scientific method, which is just not accepting what other people say, questioning people. That's all there is to it. Well, the scientific method has like 8 steps, but I can't be bothered to show it. You should remember the scientific method though. It's retaught often throughout your science courses. Next, scientist, Janssen. Around 1590, he invented the microscope, the first microscope, which only contained 2 lenses. So let's look here. This is the man Janssen. Yes. And then that is his, let's see if I can see it bigger, Zacharias Janssen, and this is his first microscope. It looks like a tiny telescope. Oops. Oh yeah, here's a picture of Aristotle, guys. Amazing. Moving on. I'm gonna get copyrighted for this. Robert Hooke. So as a reminder, you guys don't need to remember, for scientists, any scientists you learn about in science, you don't need to remember their first names. Just know their last names. So just Hooke, 1665. He studied cork under a microscope, and this is what cork looks like under a microscope. And he found that there are these things that look like compartments within the cork under a microscope, and he called the compartments cells, which means compartments. I know, the creativity is amazing. Here's an image of Hooke. He has nice hair. Next up, we have Van Leeuwenhoek. So around 1650 to the 1700s, he studied unicellular organisms, and he was the first to find about unicellular organisms. Examples of unicellular organisms are bacteria, which is a living animal made of only 1 cell. Whereas compared to us humans or animals, we are multicellular organisms. He also discovered the nuclei/nucleus. He named the unicellular organisms animalcules, which the name did not stick, obviously. Oops, why do I keep zooming in? I'm actually having issues. Fix the zooming. So here is Leeuwenhoek, Dutchman's. All right, moving on. I'm gonna start writing, because I did not finish my lesson making. So next we have Brown. His whole name, I don't remember. It's probably Robert Brown. Probably not. 1820. Let's see. I need to explain this better. All right, Leeuwenhoek. Basically, he was studying cells, right? And then he saw these dots in the cells. He saw the nuclei, but he didn't give it a name. So Brown showed up in 1820 and was like, let's call that dot nucleus. And then that's all he did. He named the nucleus. Maybe he did more than that. I'm sorry. I'm sorry, Brown, for maybe underestimating your genius. Next up, we have Schleiden in 1838. So that was about 18 years later. And he decides that, all plants are made of cells. And that the nucleus played a role in cell division, which is the reproduction of cells. Which is true. It is true, yes. All plants are made of cells. And yes, 1839, we have scientist Schwann. What he did was that he concluded all animals are made of cells. Wow. So Schwann and Schleiden, they worked together to propose, if you remember, the first law, I think, one of the laws of cell theory, one of the parts of cell theory is that all living organisms are made of cells. Next up, we have Virchow in 1855. So he concluded that all pathogenic organisms, so pathogens are just infections. They're living organisms that infect or harm a host, so bacteria or viruses. But all pathogenic organisms that he studied organisms. There we go. He studied organisms and concluded that all cells come from other cells. There you go. All cells come from other cells. This is what he did. As in, cells don't appear out of nowhere. Cells reproduce. Wow. There. Now we're done cell history. Next up, we're going to look at the two main classifications of cells. Cell classification, I should probably title it that. Cell classification. First up, here, so the two types. The two main types, there's more, but the ones you will mainly be learning about are prokaryotes and eukaryotes. Now, prokaryotes, write that. Prokaryotes. They are one of the more primitive cells. They're the first ever cells that would have existed in the start of life would have been prokaryotes. So examples are bacteria. Yeah, that's one of the only examples. Prokaryotes are unicellular. They are tiny compared to eukaryotes. They are asexual and reproduce through fission. Let's see if I can find an example of fission, cell fission. There we go. Here's how bacteria reproduce. They just split themselves in half. Here we go. Next. Asexual fission. So prokaryotes, they do not have a nucleus. All their information, so the DNA within, so normally in eukaryotes or cells, multicellular organisms, cells, all DNA or RNA is located in the nucleus. But in prokaryotes, since they do not have a nucleus, they instead have something called, I think, like a nuclear area or something. There you go, nucleoid region. And this is all their DNA and RNA. It's just kind of scattered around the cell, inside the cell. Have nucleoid region. Some prokaryotes apparently don't have a cell membrane. I have that written down here. Okay. Eukaryotes. Think eukaryotes. That's how you remember the difference. Prokaryotes are not you. They are primitive bacteria. But eukaryotes, you are eukaryote. So you. All animals are and plants are eukaryotes. Plants, animal, fungi. Fungi is kind of debatable. They are multicellular, so made of more than one cell. Each cell, like eukaryote cells, are quite large, which is 20 to 50 micrometers in size, whereas, let me come back to here, this one, they're usually 1 to 5 micrometers in size. Eukaryotes reproduce through sexual reproduction. So basically, as you know, they have sex. Humans have sex, animals have sex, plants do not have sex. They have sex, but like not sex. So some plants are asexual, but to give an example of plant, no, yeah, plants have sex. There you go. As in, if plants reproduce through the pollen of one, like bees carry pollen around, right? Think of pollen as like sperm, and then the bees carry the pollen to other flowers, and then the pollen, which is a sperm, goes into the flower, and then the flower turns into a berry, which contains a seed, and now the seed is spread and the plant reproduces. And so you can tell people plants have sex. DNA complexes. Eukaryotes have DNA complexes, which contain protein. I do not know what that means, but it is something to know. Honestly, you could pass bio without understanding anything they taught you. Just memorize everything on the page. Of course, I'm not suggesting to do that. Don't take my word, but I'm just saying. So the eukaryotes will have what is known as a true nucleus, which is just, they will have a nucleus. How do I explain this? They have a nucleus inside a nuclear membrane. So all their DNA is contained inside a ball. Whereas you could say prokaryotes have a nucleus, but they don't have what's considered a true nucleus since their DNA is not contained. And it's just kind of spread around. Most of the organelles in eukaryotes are bound by cell membranes. I just realized I did not define what organelles are, and I know I wrote the definition somewhere, and now I am going to look for it. Oh my gosh. Okay, I found my definition of an organelle. It's here. It's down here. I copied and pasted it. An organelle is a subcellular structure, so a mini structure within the cell that has one or more specific jobs to form in the cell. It's just like the organs in your body, except they are the organs of the cell. That is an organelle. Whoa. All right, coming back here. So most of the organelles within eukaryotes are bound by cell membranes. So if I were to, how do I explain that? Basically, in eukaryotes, you will have things like, eukaryotes have organelles. Prokaryotes don't. Well, they do, but not really. As in, eukaryotes have things like chloroplasts and nucleus, a true nucleus, Golgi apparatus, ribosomes, etc. You will not find those things in prokaryotes. Everything in prokaryotes is just kind of spread around. One thing you might be tested on, again, comparing and contrasting prokaryotes and eukaryotes. So I suggest remembering their differences and similarities. Next up, we're going to learn about transport within the cells. This is going really fast. So if you remember in cells, let's see, things go in and out of cells, right? Since your cell membrane is semi-permeable, and there are different ways for objects to get in and out of the cell. One way for the stuff your cell wants, for stuff to get in, is through passive transport, which happens through diffusion or facilitated diffusion. If you remember what diffusion is, it's just things moving from a higher concentration to a lower concentration. It is the movement of material across a cell membrane without the use of energy. all there is. Next, diffusion. See, normally these slides I would take much longer to go through when I wait for people to take notes, but now I'm going way too fast. Perhaps I should make you all take notes again so I don't go so fast. Molecules. Oh, I'm defining diffusion now. Molecules of a substance move from areas of high concentration to areas of lower concentration. That is it. They keep moving like that until what is known as dynamic equilibrium is achieved. I don't know. Let's see if I can find an example of dynamic equilibrium. I thought it was equilibrium. Is there some kind of animation? No, there is not. Basically, until equilibrium is achieved. Until there's an equal concentration, an approximately equal concentration on both sides. Oh, wait, I can show you stuff. So basically, if we look at, is there a large example we can see? This image is good, but it's too small. Sure, let's use this image and hope you can see it. Basically, let's say this line in the middle on the left here, on this left image model, is the cell membrane, and this is stuff outside the cell membrane. Through diffusion, there's an area of high concentration, and inside the cell, an area of a low concentration. So through diffusion, the stuff in the high concentration area moves into the low concentration area, and it keeps moving in until equilibrium is achieved. And this will keep happening because within your cell, these nutrients that move in, they get used up by organelles to make energy or produce food or to digest stuff. So later when your cell starts digesting all these things, it comes back to this again, where there's a higher concentration on the outside and a lower concentration on the inside, and the process continually happens. It is a cycle. Now, another term we need to know is rate of diffusion, which is exactly what it sounds like, speed of diffusion. Now we need to know things that affect rate of diffusion. So factors include the difference in concentration. So if, let's say, something that has a higher difference in concentration versus something that has a lower difference concentration, the thing with the higher difference concentration will have a faster rate or speed of diffusion. Next, another thing that affects it is permeability. So how easy it is for something to get into and out of the membrane or border separating the two areas of concentrations. Next up, temperature. So as you know, when something is heated up, you guys probably learned this last year, in science, when something is heated up, more energy or heat causes the cells to become excited, stuff within the body to be excited, and they start moving faster, causing a higher diffusion rate. Next, biological membranes. This is just a term which is defined as biological membranes are selectively permeable membranes, which another name for it is again just semi-permeable membrane. This is going so fast. We've gone through 50 slides. Osmosis. Osmosis is diffusion, but water. Because water is special, they get their own name for their diffusion. Yeah, that's it. That's all there is. That's the definition of osmosis. Okay. Now, because I went way too fast, this lesson happens to end early. So bye-bye, guys. I went way too fast. Next time I'll just talk slower or something. Y'all should leave comments or email me and tell me if I'm going way too fast. Bye-bye. Enjoy your Halloween. See you next week as we move on in biology.